When we talk about cell cycle is defined as the stage through which a cell passes from one cell division to the next cells. Right? Next, collectively referred to as a cell cycle. So from one cell, right, from one to become two, then two become four, another cell cycle. Okay. So timing cell cycle varies vastly, but generally about 8 to 20 hours, depends on the plants or animal cells. So the cell cycle consists of three main phases: interphase, nuclear divisions, or we know as a mitosis, and cytokinesis. Okay. So today our focus will be in interphase. Okay. So if you look at this, interphase is the longest one, green color. So we have G1 as G2. So it takes about 18 hour. Okay, talk about 18 hour, and then followed by a mitosis, cytokinesis. Okay, can you see that? Mitosis and cytokinesis. Okay, so what's the interphase? Now, a lot of times, the cells actually spend their time in the interphase. They spend the time in interphase. And at this moment, the cells actually, they are metabolically very, very active. Highlight because they are synthesizing proteins as well as the protein, lipids, and other biologically important molecules. For example, polynucleotide. Okay, during the interphase. So it means that why? DNA replication also takes place during interphase. Okay, so the time between the end of cell division and the beginning of the S phase is termed as G1. G stands for gap, not grow. Are you clear? We call it as a gap 1. So gap 1, what actually happened during this gap 1? Basically, we had the growth and the metabolic reaction take place. Toward the end of the G1, enzyme required for DNA synthesis become more active. Synthesis of this enzyme along with the protein needed enable the cells to enter the S phase. So what actually happened here is during the first G, the first gap, the cell actually prepare for the DNA replication. Okay, so a lot of protein synthesis, everything get ready for the DNA replication. So sometimes certain cells that are not dividing usually become arrested in this part of cells and said to be in the stage called G0. So it means that this kind of cells, they won't carry out DNA replication. They stop. They are arrested. Then this is very, very important. You try to imagine that, okay, if we already grow to adult, we have to arrest most of the cells. We don't want them to continue to multiply because useless, right? Mature enough already. So you don't need to get taller, you don't need to get fatter in this case. So therefore, the cells will be arrested, okay? Then after G1, you have the S phase. So S phase, the DNA replicates semi-conservatively, okay? At the completion of S phase, the cell enter the second gap phase known as G2 phase. So in this case, increase protein synthesis. So we try to make a lot, as much as protein as possible, because after that mitosis, the cells, okay, won't be able to make protein anymore. So protein only can be made during the G1 and G2, the gap 1 and gap 2. So because of G1 and G2, now the cells start to grow, and because of the certain enzyme and protein, the cells can carry out certain functions. For example, liver, they had a detoxification enzyme. Can you see that? Okay, for red blood cell, for example, then they have the carbonate anhydrase, have hemoglobin, so during the G1 and G2. Are you clear? Okay. So if you look at this, this is a typical structures of a nucleus of the cells during interphase. Okay, so what is the visible feature of the cells in interphase view under light microscope, guys? So presence of the single prominent nucleus, very prominent nucleus. And then you can see the nuclear envelope and nucleus remain intact. Nucleus remain intact, nuclear envelope remain intact. So this is the features of the, okay, the nucleus of the interphase. Are you clear? And third, you can see that no visibility of the chromosome. In this case, can I see chromosome? Let you see what is mean chromosome. Huh? This is mean chromosome. Can you see that? Individual chromosome. Can you see that? But here, can I see the chromosome? I won't be able to see the chromosome. So this one must be interphase. Okay, so highlights number 8. Point number 8. So the sometime question will ask how you know that this is the cells in interphase. So first we check. Oh, they have the nucleus there. They still have the nuclear envelope and nucleolus remain there. And then you can see that no chromosome available. So this one must be interphase. Okay. So after interphase, the cell will progress to the mitosis, which we're going to discuss it tomorrow. Okay. So any more question?